Okay, in this screencast here we're going to look at incomplete dominance and codominance, which is a little bit different than what we've looked at before. In this case we have genes where there's not a normal dominant recessive relationship. So we're not having one gene which is a lot stronger than the other, where we had a dominant gene that is expressed over the top of the recessive gene. We have a situation where these black and white genes, where we'd have a black gene and maybe that white gene, and they're both expressed equally. Now when we do this, there's two ways it could turn out. One is where it's incomplete, and this is usually best thought of as being a blend of the characteristics. And if we think about what the word incomplete means, it means it's not completely one or it's not completely the other. It's in between, so incomplete. The other alternative we have is codominance, and co really refers to it being both. Okay, both genes are expressed. And if we think about this, a blend of black and white would be a grey individual, and codominance would mean that both black and white are expressed. So you might have an individual with a black individual with white spots, or black and white stripes, or some black hair and some white hair. So that's probably important to just, if you're getting confused, to stop and think about what those words mean. Okay, let's have a look at an example here, and we're going to look at snapdragons, which are a type of flower. Not dragons, you might be a bit disappointed. And we have basically two genes involved. One gene that codes for white and one gene that codes for red. Okay. Now when we write out these genes, we write them out specifically, and because it's a situation of incomplete dominance, we have two dominant genes, so we write out the colour like this, and we say, we write it, it's called a superscript at the top. We write these genes at the top, and I suppose there's a couple of ways we could write this. You do see it written in some cases just as the genes I've written here, so they might write that same individual like that. Now that's not incorrect, and you'll see that in a lot of textbooks, a lot of cases. This is probably a bit more useful if we get into more complicated scenarios, because it indicates with the capital letter here that both are actually dominant. Okay, and if we had a different allele coming along that was recessive, we could indicate that with a small letter. So it gives you a bit more flexibility. So I suppose we prefer to use this way of writing it. But don't be surprised if you see individuals written like that as well. Okay. Now let's just have a look here at our individuals. So we've got a white individual, a red individual, and the heterozygous condition, which is a pink individual. Let's write out our genotypes of these individuals. So to be white, you'd need to have two white genes, or two white alleles. And red, you'd have to have two red, and the pink here would be one red and one white. There we go. So let's move on. We'll have a look at an example here, and we might actually cross two heterozygous individuals. So again, heterozygous, red and white, cross with a red and a white. Okay, the gametes could produce could either be red or white. And look, if you're doing a good job of your monohybrid crosses, you'll probably be fine to start doing this. Not too bad. And we transfer these down here, represent our gametes. It's really a case of making some changes when you start looking at the phenotype ratios, and the rest should pretty well be the same, some slightly different symbols. Okay, so our two individuals here, or two alleles for our individual. Okay, remembering each one of these here is getting one gene from one parent and one gene from another, so one for each. And just like we did with dominant recessive, it's good to choose one letter that you're going to write first and one that you're going to write second. Otherwise, you might get a bit confused with your heterozygous individuals. Here in my heterozygous individuals, I've chosen to write the red first and the white second, and that just makes it a bit more consistent for me. I translate these individuals now over to my genotype ratio. I'm going to have 1, which is homozygous for red. I'm going to have 2 individuals which are heterozygous. And I'm going to have 1 individual who is homozygous for white. And now the real difference, I suppose, comes down to when we write our phenotype ratio and we have 1 red, which is this guy here, 2 pink, and 1 white. Okay. So that's us. Let's have a look at another example now. Let's have a look at the cattle. And look, the picture I have of our cattle here actually has them drawn, written with these alternative symbols that I've shown you before. And if we were writing the cattle, we could write them out a bit differently. We might do that as we talk about them. So this red cow here looks a bit brown, but it's red. Would have to have the 2 genes for that reddy type coat, and the white individual here, okay, 2 for white, and roan, which is that combination of having both red and white. Now, I suppose the difference between the example with the snapdragons that we just looked at is with this example, we're having both of these alleles expressed. We have little bits of white fur and we have little bits of brown fur. Whereas if this was incomplete dominance, it'd be a blend, and the fur would turn out, you know, a colour a little bit in between. In this case, it's codominant. So we have both the red and the white being shown, both being shown, co. Now let's have a look at an example here with our roan cattle. We'll do something a little bit different. We'll have a look at a red individual crossed with a heterozygous individual. Okay. Gametes produced by this one, only the red. This one could produce red or white. Okay. As we transfer these down onto our gametes on our punnet square, using our these gametes here from this parent, these from this one here. I haven't written any along the bottom here because it would basically replicate this row. It shows the possibilities. It wouldn't be incorrect to do it. It just mean we'd probably simplify our problem a little bit later. So let's go write these out. Okay, so we're going to have a red individual. Okay, gamete from here, and a white individual. Gives us a genotype ratio of 1. Oops, that looks a bit like a G. Just try that again. 1 red individual, which is homozygous red, and 1 heterozygous individual with 1 of each of the alleles. Phenotype ratio, we would have 1 red cow to 1 roan cow. Okay, and you know, we could, like we were talking about in our last episode, we could have either this written as 50-50, okay, or you know, the probability is a half of each. So we can work in either of those sorts of ways. So that's incomplete dominance and codominance. Again, go out and do a lot of problems. Good to practice.